Assalamu alaikum friends. I hope so that you all will be fine. Friends, in this video I am going to discuss about glycogenolysis. In my previous video I have already discussed about glycogenesis. If you are new, then kindly see that lecture and come on that lecture. Okay? We will cover this topic under the heading of definition, site, and the steps of glycogenolysis. So let's start our lecture. Glycogenolysis means the breakdown of glycogen. The breakdown of glycogen to formation of glucose. This is the definition of glycogenolysis, the breakdown of glycogen to the formation of glucose. Now coming toward the site for the process of glycogenolysis. Okay, site. The glycogenolysis process takes place in liver and muscles, and the important organelle that required that is cytosole. Okay, now friends, this glycogenolysis process occurs in fasting condition, and one of the most important enzyme that released from the pancreatic islets of Langerhans, that is glucagon. And glucagon hormone, it releases from the alpha cells of the islets of Langerhans. Glucagon, it is responsible for the breakdown of glycogen to glucose. And in glycogenolysis, one of the most important enzyme that performs work, and that is glycogen phosphorylase. Okay, so let's see. Now coming toward the steps of glycogenolysis. So this is the glycogen molecule. Here is the alpha 1 4 glycosidic bond, and this is alpha 1 6 glycosidic bond. This is the glycogen molecule. Okay. Now here, one of the most important enzyme that acts, and that is glycogen phosphorylase enzyme. Glycogen phosphorylase enzyme. This enzyme, it is responsible for the successive removal of glucose molecule until it leads to the formation of limit dextrin. Okay, limit dextrin. Now this is the formation of limit dextrin. See, a limit dextrin, it is the hydrolyzed form of this glycogen. It has 4 glucose residues. See, It has four glucose residues on each side. One, two, three, four. These four glucose residues. From here, there are four glucose residues. From upper side, there are four glucose residues. It is limited dextrin. It is the hydrolyzed form of the partially hydrolyzed form of glycogen, that is limit dextrin. It has four glucose residues on each side. Okay. Now here, the glucose molecule they release in the form of glucose 1 phosphate. With the help of mutase enzyme, it converts into glucose 6 phosphate. Now, glucose 6 phosphatase enzyme it is absent in muscles. So, in muscles, this glucose 6 phosphate directly enters into glycolysis process. But in liver, it converts into the glycolysis process. It converts into the it converts into glucose with the help of glucose 6 phosphatase enzyme, because this enzyme it is present in liver and kidney. And in liver, with the help of glucose 6 phosphatase enzyme, this glucose 6 phosphate converts into glucose, and ultimately in liver it leads to the process of glycolysis and Krebs secondary electron transportation. While in muscle, this glucose 6 phosphate it directly enters into the glycolysis process due to deficiency of enzyme, that is glucose 6 phosphatase enzyme. Okay, this is the limit dextrin. Now, in next step, there is the action of very important enzyme, that is debranching enzyme. That is debranching enzyme. Listen very carefully, friends. This debranching enzyme it is bifunctional. It catalyzes two reactions. Now let's see the first reaction. The first reaction is the transferase activity of debranching enzyme. What? Transferase activity of debranching enzyme. Now what happens? Okay, transferase activity of debranching. What happens? That there is that three glucose molecule. These three glucose molecule, they transfer from upper side to this side. With the help of transferase enzyme, these three glucose molecule they transfer from this side to this side. Okay. Now I'm writing here. This is the glycogen. One, two. Okay, okay. I'm writing with the another color, that is okay. Now there is a transfer of these glucose molecule from this side to this side with the help of transferase activity. Okay, with the help of transferase activity. I am writing here transferase activity. Okay. Now this is the first action of this debranching enzyme. In next reaction, with the help of alpha 1 6 glucosidase activity of this enzyme, this glucose molecule it gets released. I am writing here. With the help of alpha 1 6 glucosidase activity, this glucose molecule gets released. Okay. With the help of alpha 1 6 glucosidase activity. Okay, so these two reactions they are catalyzed by debranching enzyme. First one is transferase activity and another one is alpha 1 6 glucosidase activity. Now there is a formation of linear chain of glucose. Okay, this is the linear chain of glucose molecule. Now Here there is a function of again glycogen phosphorylase enzyme onto the linear chain of the glucose molecule. We can say linear chain of glycogen. There is a function of an enzyme, that is glycogen phosphorylase, and it converts this linear chain of glycogen into glucose 1 phosphate. Okay, I'm writing glucose 1 phosphate. And in next reaction, this glucose 1 phosphate, it converts into, with the help of phosphoglucomutase enzyme, with the help of mutase enzyme, glucose 1 phosphate, it converts into glucose 6 phosphate. Okay, and now in liver, in liver, this glucose 6 phosphate, with the help of glucose 6 phosphatase enzyme, it converts into glucose. While in muscle there is deficiency of glucose 6 phosphatase enzyme, so in muscles this glucose 6 phosphate directly enters into the glycolysis process. So this glucose 6 phosphate in muscles, this glucose 6 phosphate directly enters into the glycolysis process in muscles. Okay, I'm writing, sorry, here in muscles, this glucose 6 phosphate directly enter into the glycolysis process. While in liver, this with the help of glucose 6 phosphatase enzyme, glucose 6 glucose 6 phosphatase enzyme, it converts into the ultimately glucose in liver. In this way, the glycogen breaks down into the glucose. I am repeating that this is the glycogen molecule. Okay, with the help of glycogen phosphorylase enzyme, it is responsible for the removal of successive glucose molecule until it leads to the formation of limit dextrin. These glucose molecule, they release in the form of glucose 1 phosphate. With the help of mutase enzyme, they convert into glucose 6 phosphate, and ultimately in liver, glucose 6 phosphate enters into the, in liver. In liver, it converts into the glucose, which enters into the glycolysis process, Krebs cycle, and electron transport chain. While in muscles, this glucose phosphate directly enters into the glycolysis process due to deficiency of glucose 6 phosphatase enzyme. This limit dextrin having 4 glucose residues on each side. This is a limit dextrin and it is a partially hydrolyzed form of the glycogen. With the help of debranching enzyme, it has two activity, that is transferase activity, and another one is alpha 1,6 glucosidase activity. With the help of this activity, these three glucose molecules, they transfer from here to here. And you can see this is the transferase activity of the debranching enzyme. And next, this with the help of alpha 1,6 glucosidase activity, there is removal of this glucose molecule. There is a removal of this glucose molecule, and this glucose molecule gets released, and we there is a formation of linear chain of glucose or linear chain of glycogen. Okay. With the help of glycogen phosphorylase enzyme, there is the formation of glucose 1 phosphate. With the help of mutase enzyme, this glucose 1 phosphate converts into glucose 6 phosphate. Now, in liver, with the help of this enzyme, glucose 6 phosphatase enzyme, this glucose 6 phosphate converts into glucose. While in muscles, it directly enters into glycolysis process due to deficiency of glucose 6 phosphatase enzyme. So friends, it was all about glycogenolysis. If you like this video, then kindly subscribe to my channel and share it. Keep watching and thank you.